In this lecture, we will discuss type 2 alveolar epithelial cells and pulmonary surfactant. This topic is essential for understanding normal lung mechanics as well as diseases such as neonatal respiratory distress syndrome and acute lung injury. Identity of type 2 alveolar cells. Type 2 alveolar epithelial cells are visually conspicuous cells. Although they are commonly referred to as large alveolar cells, their mean volume is actually less than half of type 1 cells. They appear large mainly because of their cuboidal shape and prominent cytoplasm. Location and distribution. Type 2 cells are preferably located at the corners of alveoli and near interalveolar pores of cone. Under normal conditions, they are usually solitary, but during epithelial injury, they proliferate and form focal clusters. Importance of apical surface specialization. The apical surface of type 2 cells bulges into alveolar lumen and shows numerous short microvilli. These microvilli increase surface area and are related to secretory function of the cell. Junctions with type 1 cells. Type 2 cells form junctions with neighboring type 1 alveolar epithelial cells. These junctions are important for maintaining epithelial continuity and overall barrier integrity. Cytoplasm of type 2 cells is rich in organelles including mitochondria, rough endoplasmic reticulum, Golgi complex, and lysosomes. This reflects their high metabolic and synthetic activity. Lamellar bodies. The defining ultrastructural feature of type 2 cells is the presence of lamellar bodies. These are large membrane-bound secretory organelles containing densely packed phospholipid lamellae and are among the largest secretory organelle in the body. Lamellar bodies as secretory lysosomes. Lamellar bodies contains lysosomal enzymes, express lysosomal membrane proteins, and have an acidic pH of around 5.5. Hence, they are regarded as secretory lysosome-related organelles. Major function of type 2 cells. Type 2 alveolar epithelial cells have two major functions. They are the cellular source of pulmonary surfactant and they serve as progenitor cells for regeneration of alveolar epithelium. Composition of pulmonary surfactant. The pulmonary surfactant consists predominantly of lipids, mainly saturated phosphatidylcholine, and smaller fraction of surfactant proteins. Functions of surfactant proteins. SPA and SPD are hydrophilic proteins involved in innate immunity, whereas SPB and SPC are hydrophobic proteins essential for surface film stability. Synthesis of surfactant. Surfactant synthesis involves coordinated lipid and protein production, processing in Golgi complex, and storage in lamellar bodies. Secretion of surfactant. Surfactant is secreted by exocytosis. Lamellar bodies fuse with apical plasma membrane, forming a fusion pore through which the surfactant is released. Tubular myelin formation. After secretion, lamellar body contents transform into tubular myelin, a lattice-like structure located in hypophyse, and this process requires SPA and SPB proteins. Surface filling. Tubular myelin serves as a precursor for surface film, which lines alveoli and reduces surface tension, preventing alveolar collapse. Surfactant recycling. Major route of surfactant clearance is by reuptake through type 2 cells. Within type 2 cells, they are either recycled or degraded. Minor clearance occurs via alveolar macrophages and the airways. Surfactant pool. Surfactant exists in multiple intracellular pool as lamellar bodies and intra-alveolar pool as lamellar body forms, tubular myelin, surface film or unilamellar vesicles reflecting different functional and metabolic states. Surfactant turnover. Surfactant turnover is rapid with a turnover time of 4 to 10 hours. Because reserves are limited, lung has reduced capacity to cope with sustained injury. Mechanism of surfactant dysfunction. Surfactant dysfunction may result from impaired synthesis, cellular injury, or inactivation within alveolar space. Clinical correlation. These mechanisms explain the role of surfactant dysfunction in neonatal respiratory distress syndrome, acute lung injury, and interstitial lung diseases. Therapeutic implications. Understanding surfactant biology has led to surfactant replacement therapy, a major success in neonatal medicine, and variable benefits in adult lung diseases. In summary, type 2 alveolar epithelial cells are central to surfactant production and alveolar repair. Lamellar bodies are key to surfactant storage and secretion, and surfactant is essential for normal lung function and alveolar stability. This diagram summarizes the pathway of pulmonary surfactant synthesis and secretion by type 2 alveolar epithelial cells. Surfactant components are synthesized in endoplasmic reticulum, processed through Golgi complex, stored in lamellar bodies, and released by exocytosis into alveolar lumen, where they form tubular myelin and surface film. This diagram is shown here as a visual reference to reinforce the concept discussed earlier.